All right, so now is in the first part we discussed about the DNA replication introduction. Right, so now we'll see how the DNA replication happens. Right, so the DNA replication that is done by the replication machine, right? By initiated protein binds to a specific DNA sequence called replication of origin, right? Replication of origin, the initiated protein binds to the supposed replication of origin and then it becomes replication fork. So first, when you think about DNA replication, so which enzyme is responsible for replication of DNA is DNA polymerase. And this enzyme is responsible for the polymerization reaction in the DNA replication. Correct. So now, right. So I will make a image of a DNA, just a simple 2D image. All right, maybe like this. Right. So this is our, for example, C, right, and here we have G. All right, so these are our bases that I'm just putting an arrow to it. C, that is C, and maybe this is T, and just a random arrows for the DNA, right? So this is the 5 and 3N, and this is a 3 and 5N, right? So this is, for example, our basic DNA structure. So when the polymerization happens, so like just imagine that this is the structure that we are having and this is open from here. So the DNA replication is happening over here, and this DNA polymerase is responsible to add a new nucleotide to this end, right? So I will write here adding new nucleotide right here at this end. Oops, I think I should write, and this is, I will make this long, for example. Right, this is G and maybe oops, this is T. All right. So the DNA polymerase will add here new nucleotide. So the new incoming nucleotide for the polymerization reaction that will enter this reaction here as a deoxyribonucleoside triphosphate. So I will write it down here. New incoming nucleotide will enter as deoxyribonucleoside triphosphate, right? So it will enter this reaction as a deoxyribonucleoside triphosphate. And what is this? So you can see an image of deoxyribonucleoside triphosphate. So when we see here, this is a nucleotide, right? With just one phosphate, then deoxyribonucleoside, then deoxyribonucleoside diphosphate, right? This goes on, and then deoxyribonucleoside triphosphate having 3 phosphate groups with this, right? So this incoming will occur as, for example, like, so I will use something else. Alright, so for example, this is our incoming, and we'll make like this with 3 P's, right? So deoxyribonucleoside triphosphate is coming here. So this enter the reaction as this. So the energy for this polymerization reaction is actually provided by this incoming deoxyribonucleoside triphosphate itself. How? So when the hydrolysis of one of the high energy phosphate bonds will happen, bonds fuse this reaction. So like here we have, so we just need one with the P, right? Like here, it should join with just one phosphate, and so it will remove this too, the hydrolysis of this phosphate, one of the high phosphate bond, and by releasing the pyrophosphate. So that provides the energy, and the pyrophosphate, this pyrophosphate further hydrolyzed to inorganic phosphate, right? So this is how the reaction helps, that energy helps this reaction, and eventually that we have our new, right, nucleotide, that will be C, right? So this will, the all the incoming nucleotides, so the DNA polymerase will make sure whatever nucleotide is incoming, it will try to match, or it will match, not try, it will match this bases. So as we know, G with the C, T with the A, right, so on, is the same. So whenever, for example, here is T, so whenever the new nucleotide is coming, so the DNA polymerase will see, okay, we have T, so now we have to put A to match with the other strand of the DNA, right? So this is what it happens. All right, so this what happens. So DNA polymerase, right, responsible. So I can draw like here a shadow of a DNA polymerase, maybe like this. That is the DNA polymerase acting, and we'll name DNA polymerase, right? So the DNA polymerase is acting on this DNA and adding a new nucleotide at this end, right? So the replication fork, as we know, this is a replication fork, right? And both has a different direction. This goes this direction, this goes this direction. So for example, this right-handed replication fork we take, so the DNA replication that is happening in this right-handed DNA, right? So I will just make here, right? So this is our, this is going here. We have this 3N, 5N, and 5 and 3N, right? So the replication fork is going on this way, right hand side, and the DNA polymerase, so that replicates the DNA only in one direction, that is just this direction, 3 to 5, right? If it is using this template, so it will replicate in 3 to 5 direction. So what happens to this 5 to 3, right? If this, it is using this 3 to 5 as a template, so what about the above one, right? So as I mentioned, so the new DNA replication that is happening, it happens in just 3 to 5 direction, right? So it makes this strand, but the DNA replication on the other strand is being made, it's like this 5 to 3, so it's not, so this is going this way 3 to 5, so other is not going in this direction, opposite direction. The DNA replication goes in just one direction as the replication fork movement, the same as, so if this replication fork is going in this way, so the DNA polymerase will go just in this way, it won't go backward for like the direction of replication fork. All right, so write down direction of replication fork and DNA replication is the same, right? It will be the same. If this goes 3 to 5, this goes on this way 3 to 5. DNA replication will be done in replication goes in in 3 to 5 direction and this direction. So what happens to this above? So it's not like there are like two DNA polymerase that are one is going on this way and one is going on that way. No. So that is just one single DNA polymerase that is acting on the 3 to 5 direction of the replication fork. So what happened is that the DNA polymerase uses a back stitching method. So DNA polymerase using back stitching method. If you know how to sew, you will know back stitching method, like in the sewing that is used. So what happens, how this happens, I will show you. I will use the same thing here. All right, so this is 3 to 5, this is 5 to 3, right? One the replication fork is going this way, one going this way, right? So the new DNA that is formed on this replication fork will be right 3 to 5 direction this way. So when the DNA polymerase will add the new nucleotide to this, so what happens? So for example, I'll show you. For example, this is the halfway. I will show you. This is a nucleotide, the new one, pink. It's the new strand. Oops. All right, so this is the new DNA strand that is happening. Right, so DNA polymerase is going here. It drops. It goes on just this direction, dropping new nucleotide just to 3 to 5. But what is happening on this 5 to 3 direction? So new one is dropped here, right? And I will make a small DNA polymerase. All right, new one is dropped here. Then new one is dropped here, and the DNA polymerase is moving. But what it does, so when DNA polymerase do this one section, for example, I should show you maybe. Right, so how it does the back stitching method. Okay. Let's do it here. Oops. All right. So 3, 5, 5, 3. This replication fork this way. This goes this way, right? So we have this replication. The new DNA that is being made. This is here, right? That both on this side. So what the DNA polymerase does. So for example, this is our DNA polymerase, right? So DNA polymerase is adding new new nucleotides. So here in the 3 to 5 direction, right? DNA polymerase, add new nucleotide, right, to 3 to 5, right? So what happens to this 5 to 3 direction? So, and the DNA polymerase moves on this direction, right? It is going in this way. So, all right, so I will make one more, or else I will just make this part. right? Just this section over here, right? 3, oops, sorry, 5. So what is happening is this 5-3 direction, how it's made. So for example, this has dropped a new nucleotide, then the DNA polymerase moves further, and for example, this is where we have our DNA polymerase, right? So it is adding the new nucleotide and making the new DNA strand over this. But here, so as it goes one further, it slightly comes back and drops a nucleotide. Again, it continues making here at the bottom in the 3 to 5 direction, but it goes further, it slightly comes back as in the same as a backstitching method and drops a nucleotide. Slightly goes there, comes back and drops a new nucleotide. And so this fragments is actually called a fragments. It is dropping the fragments and it's fragments together, and then it attaches. This attaches to this 5 to 3. That is not this one. This is the new process. But like for example, if we have this as a strand or as a template, right, and the new strand will be, so this is dropping. It's called an Okazaki. So this is called Karzaki fragments. The DNA polymerase goes in this direction, and then so it goes one, moves slightly back, drops a DNA fragment, goes further, drops another DNA fragments, right? Then that is called an Okazaki fragment. This is named after the biochemist who discovered them, and these are later joined together to form a continuous strand. So this is how on this side, 5 to 3 side, this is how the new DNA strand is made. So this strand that is going here is called a leading strand. And the one that is made by the backstitching method is known as a lagging DNA strand, right? So this is what is happening, the DNA replication, right? So we have a leading strand and a lagging strand, right? So DNA, this is the leading strand, right, where the DNA polymerase is moving further, and this is a lagging strand where the DNA polymerase drops the DNA fragments, that is the Okazaki fragments, and later it's joined to form a new DNA strand. And why does this? So this replication fork of all cells, prokaryotics or even eukaryotic, have a leading and a lagging strands. And this common feature arises from the fact that all the DNA polymerase work only in one direction, that is 5 to 3, right? 5 to 3 direction, a restriction that provides cells with an important advantage. Right? Why 5 to 3? Because so this is the 3 to 5 and the new DNA strand that is made is 5 to 3. That's why. And so the advantage why this DNA polymerase does this. Right? So why the DNA polymerase goes in this direction? Because DNA polymerase need to proofread the nucleotide that are attaching to the new strand. So the DNA polymerase is so accurate that it makes only about one error in every 10 raised to 7 nucleotides per it copies. So that is the error ratio of a DNA polymerase. So this error rate is much lower than can be explained simply by an accuracy of a complementary base pairing, right? So the base pairing is GC80, right? This is the base pairing. But although the AT and CG are the far most stable base pair, so these are the very stable base pair, There are other less stable base pairs are as well, such as G and T or A and C, right? Can also be formed, right? So DNA, when the DNA polymerase is adding a new nucleotide, so this base can also be formed like C and T or A and C, right? This can also be formed, but the error ratio is very less. So such incorrect, so whenever such incorrect base pairs are formed, right, much less frequently than the correct one, but if allowed to remain, if that is allowed to remain on the DNA strand, they would result in the accumulation of mutations. So this disaster is avoided because DNA polymerase has two special qualities, right? That greatly reduces or greatly increases rather the accuracy of DNA replication. So first, the enzyme carefully monitors the base pairing. So while it is making the new DNA strand, it carefully monitors the base pairing. Like here, this is T, so there should be a that is monitored, right? And the template strand, right? This, and only when the correct match is available, the DNA polymerase catalyzes the nucleotide addition reaction. If it's A and it's T, it makes sure then only it catalyzes the reaction. If it's T and if it's here G, it won't catalyze the reaction. That it makes sure, right? And when the DNA polymerase makes a rare mistakes and adds a wrong one, for example, nucleotide, it can correct through an activity called proofreading. So what does the DNA polymerase do? It adds, it first makes sure it's T, so it's need an A, right? But for example, by mistake, if T goes with C, then whenever after putting the new nucleotide, DNA polymerase proofread. Alright, so this is what T, I need A, it makes sure first, then after the reaction is completed, it's still proofreaded. Okay, so this is T and I put it here A. So that is correct. If it proofreads and it says, okay, it's T and I just put it G, it's not correct. So it can remove that nucleotide by the nucleus, right? That is responsible for breaking the bond between the two nucleotides, right? So proofread takes place at the same time as the DNA synthesis. So this proofreading is at the same time as the DNA synthesis. Before the enzyme adds the next nucleotide to a growing DNA strand, it checks whether the previously added nucleotide is correctly base paired to the template strand, right? So this proofreading is carried out by a nucleus, right, that cleaves the phosphodiester backbone if the DNA is wrong, or sorry, if the nucleotide is wrong. And so the polymerization, adding of the nucleotide and the proofreading are tightly coordinated, and the two reactions are carried out by different catalytic domains in the same polymerase molecules. Right, so this polymerase has both the catalytic reaction for adding as well and for cutting as well. Right, so that was DNA polymerase and the DNA replication process, but this is just a part of a DNA replication. There is much more to DNA replication. Okay, so we'll discuss in the next video the RNA, right? It's actually responsible with the DNA replication. So we'll see you in the next video. Thank you guys for watching, and don't forget to subscribe.